Welcome to Cell Biology. Today, we are diving into the fascinating world of protein sorting and transport. Imagine a massive, bustling city with millions of packages being delivered every single second. Your microscopic cells do exactly this, ensuring every newly built protein reaches its precise destination to keep you alive and healthy. To begin, let us understand why this matters. If highly destructive digestive enzymes accidentally end up in the cytoplasm, they would digest the cell from the inside out. To prevent this, the cell uses a brilliant system called the signal hypothesis. Every newly built protein has a built-in molecular zip code called a signal sequence. As the protein is being made, a cellular postman, known as the signal recognition particle, binds to this sequence. It temporarily halts production and drags the entire complex to the endoplasmic reticulum, ensuring the protein is safely threaded inside. Now that we understand what the signal recognition particle does, let us look at the mathematics of how tightly it holds on to its target. This formula calculates the dissociation constant, Kd. Conceptually, it measures the binding strength between the protein and the particle. P represents the concentration of the free protein, and S is the free signal recognition particle. C is the concentration of the successfully bound complex. If the binding is very strong and efficient, the complex forms easily, and the value of Kd decreases. Conversely, if a mutation makes the binding weak, Kd increases, and the protein might fail to reach its destination. Before we move on, let us see this with a concrete example. We are given the concentrations of the free protein and the free particle, both in the numerator, and the bound complex in the denominator. We want to find the dissociation constant to see how efficient the cell is. First, we multiply the free concentrations together, giving us 10 times 10 to the negative 12, which simplifies to 1 times 10 to the negative 11. Then, we divide this by the bound complex concentration. Subtracting the exponents gives us a final Kd of 1 times 10 to the negative 6 molar. This small value physically means the cell is highly efficient at capturing proteins. This formula we just used leads us to an important question. What happens during a genetic disease? Here, a mutation increases the dissociation constant to 5 times 10 to the negative 5. We need to find the new concentration of the bound complex. We rearrange our formula to solve for C by multiplying and dividing the respective terms. Plugging in our values, the numerator remains 1 times 10 to the negative 11. Dividing this by our new, larger Kd yields a final bound complex concentration of 2 times 10 to the negative 7 molar. Physically, this massive drop proves the mutation strands crucial proteins in the cytoplasm, causing disease. Now that we have calculated the binding efficiency, let us visualize this entire targeting mechanism. In this diagram, you can see the large blue ribosome translating the messenger RNA strand. The newly forming protein is emerging, and its N-terminal signal sequence is immediately grabbed by the signal recognition particle. Notice how the particle escorts the whole machinery down to the lipid bilayer of the endoplasmic reticulum. It docks perfectly with the SRP receptor, acting like a lock and key. This allows the protein to be threaded directly through the membrane channel into the membrane channel, into the lumen, keeping the rest of the cell safe. So we have successfully targeted the protein to the endoplasmic reticulum. This brings us to the next logistical challenge, moving it to its final destination. The cell cannot just teleport proteins. It uses an active energy-consuming process called vesicular transport. Proteins are packaged into tiny spherical membrane bubbles called vesicles, which act as microscopic delivery trucks. Specialized coat proteins physically bend the membrane to pinch off these spheres. This process is the absolute lifeline of cellular communication. For instance, every time you eat, your pancreas uses this exact mechanism to secrete insulin into your bloodstream to regulate your blood sugar. Now that our vesicle is traveling through the cytoplasm, it must know exactly where to unload its cargo. This precise docking and fusion process is controlled by complementary proteins called snares. A vesicle snare physically intertwines with a target snare on the destination membrane. They twist together like a tight molecular zipper. This zippering action is so forceful that it forces the two lipid bilayers to merge, releasing the protein cargo inside. Understanding this is crucial for medicine because deadly toxins like Botox and tetanus work specifically by destroying these snare proteins, completely paralyzing vesicular transport. To understand how snare proteins force membranes together, we must look at the thermodynamics driving the process. This equation calculates the Gibbs free energy change, delta G, which tells us if fusion happens spontaneously. The standard free energy change, delta G naught, represents the inherent energy of the snare's twisting. R is the gas constant, and T is the absolute temperature. The fraction represents the concentration of fused membranes over the separate vesicle and target membranes. When snares zipper, they release massive energy, making delta G highly negative. This physically forces water out of the way, driving the membranes to fuse. Let us apply this thermodynamic equation to a real biological scenario. We want to calculate the energy released when a synaptic vesicle fuses to release neurotransmitters. We are given a standard free energy of negative 30,000 joules per mole, body temperature of 310 Kelvin, and a concentration ratio of exactly 1. We substitute these values into our equation. Because the natural logarithm of 1 is exactly 0, the entire right side of the addition cancels out completely. This leaves us with a final delta G of negative 30,000 joules per mole. This large negative value means the snare proteins act like a loaded spring, snapping shut to release neurotransmitters instantly. Now that we have seen a single fusion event, let us see what happens when many vesicles have already fused. The concentration ratio is now 100. We use the exact same Gibbs free energy equation, but this time the natural logarithm of 100 evaluates to approximately 4.605. Multiplying this by the gas constant and temperature gives a positive thermodynamic resistance of about 11,868 joules per mole. Adding this to our standard free energy gives a final delta G of negative 18,132 joules per mole. Physically, this means the buildup of fused membranes acts as a natural braking system, preventing the cell from expanding too rapidly. Having calculated the energy of fusion, let us look at the physical structures that make it happen. In this diagram, you can see the transport vesicle approaching the target lipid membrane. Notice the red V snare proteins on the vesicle and the blue T snare proteins on the target. As they make contact, they form a tight snare complex, zippering together. This mechanical twisting provides the exact energy we just calculated, pulling the membranes so close that they seamlessly merge into one continuous lipid bilayer. The blue protein cargo is then successfully released into the new compartment. We have covered the full journey of a protein today. So let us review the most important ideas. First, the signal hypothesis explains how built-in amino acid zip codes and the signal recognition particle direct proteins to the endoplasmic reticulum. Second, vesicular transport acts as the cell's microscopic delivery system, moving these packaged proteins between organelles. Finally, the critical step of delivering the cargo is powered by the energetically favorable zippering of snare proteins. You now understand the elegant, energy-driven mechanisms that ensure cellular communication and keep our bodies functioning perfectly. Great job following along.